Okay, so first I give you an overview of the website. Second, I told you how to use site search. And now I'm going to tell you how to use the search strategy system. So if you come to the website, it could be VectorBase or the central viewpad site or any of the other sites, you're going to find the strategy system here on the side where it says search for, and you can also find it here at the top where it says searches. Either way, it's going to be the same. And you notice here, there is different categories, genes, organism. You can select one of those, for example, expand all. I will suggest you expand all because it's the first time you are going to use it to give you a general idea of what are the types of in silico experiments that you can run. And once you select on a heading and a subheading, it will actually give you the list of available strategies that we have available for you. And there are many. So for that reason, even though if you take a look of the categories, you're not going to remember them all. So that's why we have here this box that says filter the searches below. And you just type in keywords, for example, transmembrane domain or gene ID, and I will show you only the strategy that you are interested in. And this is how a strategy will look once you complete it. In the exercise that we created especially for you is going to give you the step-by-step how you create this step exercise, this step strategy system. So for example, and you will learn how to give them a name, how to save them, how to copy them, how to edit. So once you, for example, as soon as you create it, you remember what you did in each one of the steps, but maybe you probably are going to forget. So the same way you keep records in your notes when you go to the field, or you have a lab notebook for the experiments you do in the lab, this is good also to keep records of how do you do bioinformatics experiments. So here you can write what did you intended with running each one of these steps, and you can include more steps clicking here and add a step. Once you select, for example, here I selected step number 4, and it's telling me I started with 122,000 genes for Culex quinquefasciatus. I was looking for the genes that had a transmembrane domain. And later, I gave a list of a specific interpro domain. So I have a list of genes here and a list of genes here. And this symbol is representing that I'm doing an intersection from the genes I have here and I have here. It's like if you have an Excel file and you have column A and column B, and column C is going to be what is common between those two columns. That's exactly what I did here. So between with those 233 genes, I go to step number 3. And here I selected organisms. For example, I said, oh, my organism of interest is this mosquito, Culex quinquefasciatus. And later I said, well, I want to find, for example, the orthologs in those genes for a specific species. And that's why I end up with 6 genes, even though I started with so many, because I refine my search to look what I want to find. And here I select, and notice this is the only step that is yellow, and it says 6 genes. And in the table below, I have the 6 genes that I am interested. You have all these different columns available, and you can click here and add columns to add more, or you can close the ones that you are not interested. So I give you an overview of how a strategy can be started and how does it look at the end with all the steps and how can you look at the list of genes that you obtain. Now I'm going to give you the step-by-step of actually how do you construct the strategy. So remember what I told you previously is that you can start a strategy from here, from the searches menu, or here from search for. You need to remember what are the different categories and subcategories to start your strategy. Just type a keyword. In this case, I type inter because I want to work with inter-pro domains. I select that type of search and just start filling all the fields. So first is asking me which species or organism that I want. I want to select all the arthropods. Which domain database I want to use, interpro scan. And for the specific domains that I want to search, I did my homework first, and I want antimicrobial peptides, and I search for the specific IDs. But if you don't want to do your homework first, you just start typing, and autocomplete will give you what you need. And once you are satisfied with all the fields, you just click on Get Answer. And notice here, I have my first step. I can rename my strategy. And right now it only has one step, and it says I have 321 genes, and that's what I have there. Add a column, as I told you before. Type a keyword. You don't need to expand all the available possibilities. Protein length, for example. Gene name or symbol, just with the few letters at the beginning, autocomplete would suggest for you, and we'll add those columns to your table. Notice some of the genes have an unspecified product, and those do not have a gene name or symbol. I can expand because remember I selected all the arthropods, so I can have a quick overview of how many genes for my query are in each species. For this Egypti, I have 12, for example. For some species, 3, 9. So for exactly the same query with the same protein domain, I have a different number of genes. And apply my filter, and I only want to take a look of the genes for Aedes aegypti, and that's why I have this symbol here, and I'm only looking at the 12 genes for Aedes aegypti. It's the same query, but I just filtered to see that. Add a step is what it will take me to step number 2. Now what I wanted. I want a predicted signal peptide because I want proteins that are secreted. Again, I need to tell it from which species I want all arthropods, and I run a step. Now, notice that step 2 is highlighted in yellow, and that's the list of genes that I have below. But if I click back in step 1, that becomes highlighted, and those are the genes that I have below in my table of results. Again, doing the same thing. And the operator that I did was an intersection between the signal peptide and the interpro domain to obtain the genes that are in common between those two. I'm looking at the number of genes and transcripts for each one of those queries. I can copy, I can edit details, I can save. And with save, you can give the name that you want. I recommend you use that because later with all the strategies available, you're not going to remember which one is which. And here is what I told you, you can name what you did in each one of your steps. And that, if you tick that, you make that strategy public. So step number 1, I look for 7 interpro IDs for antimicrobial peptides, and I write which peptides those are. Step number 2, which one of those genes are secreted, and that's why I selected the signal peptide, and I update that. And these are the public strategies available in VectorBase. And you can make public the ones that you construct as well.